All right.
Great, excellent. So, welcome back, everybody. This is our next talk now, and it is my distinct pleasure to introduce Kareem Zaghloul, who's
Sorry, Kareem,
Zaghloul
you have a bit of echo.
Oh, there's a little bit of echo?
Yeah.
Okay. So Dr. Zaghloul is a senior investigator in the Functional and Restorative Neurosurgery Unit at the National Institute of Neurological Disorders and Stroke. Dr. Zaghloul received his bachelor degree from MIT, his MD and PhD degrees from the University of Pennsylvania. And his graduate work focused on developing silicon models of visual processing in the mammalian retina. He also completed a residency in neurological surgery and postdoctoral research investigating the neural correlates of human memory encoding, decision, and reward. His lab is focused on investigating the neural mechanisms underlying human cognitive function. And I'm very happy that we were able to have Dr. Zaghloul with us today. And so the title of his talk here is Neural Signatures of Memory and Information in the Human Brain. So without any further ado, I will just please join me in welcoming Dr. Zaghloul.
Great. Thank you for the very nice introduction. There's a really bad echo. Can you guys hear okay?
Yeah, but there's a bit of an echo.
Do you have two Crowdcast windows open? Maybe you need to close one of them.
Yeah, let me see. Oh, yes. Okay. And are you still seeing my shared screen?
Yeah.
Okay, great. Thank you for
a
very nice introduction, Kareem, and sorry for the technical difficulty. Thank you for inviting me to speak, and thank you for giving us the opportunity to present our work. I'm actually very excited about this meeting, and I'm looking forward to the rest of the talks and to the panel discussion. So our work in our lab is largely focused on understanding the neural mechanisms that underlie human episodic memory formation. Obviously, we study memory because we think memory is both quite interesting and quite important. We rely upon our memories for our everyday experiences, not only to form our own personal identities, but we also rely upon our memories to guide and to plan our future actions. But one of our other motivations for studying memory is that we believe by studying memory, we can actually gain insights into how the brain actually represents, processes, and encodes information. And so what I thought I would do today is share with you some of our work related to memory and understanding these signals in the brain to underlie memory, and argue that we can use this to begin to gain sort of some knowledge about how information is represented in the brain. So, of course, there are a lot of different types of memory that one could study. The memory that we focus on in our lab is a specific type of memory known as episodic memory. So this is the memory of episodes or events that you've experienced at a particular time or place in the past. So this would be the memory, for example, of your first date or your last vacation. And it's argued that the study of episodic memory is really best accomplished in humans. Humans are unique in their ability to use language, and so they can use language to explicitly describe a recollection, which is one of the key requirements in the study of episodic memory. Now, the study of episodic memory, of course, is an active area of research, and it seems to involve a complex interaction among multiple structures in the brain. So when you first experience an episode or an event, you process information about the episode itself, as well as the surrounding time and place during which that event occurred in these cortical sensory areas and these cortical association areas. This information about the particular contents of an episode, as well as its surrounding context, is conveyed down to medial temporal lobe structures, such as the entorhinal cortex and the parahippocampal gyrus, and then ultimately to the hippocampus. And one of the main purposes of the hippocampus, as James just talked about, was actually establishing these relational associations between these different elements that comprise every event or every memory that we're trying to encode. The hippocampus in turn then communicates back with these medial temporal lobe structures, with these cortical association areas, and also with the prefrontal cortex. And all of this allows us to encode new events into memory and to organize these memories. Now, there's some evidence that suggests that when we actually retrieve a memory, we actually replay some of these patterns of neural activity, and that's what I'm going to talk about today. So the framework that we use to guide our work is pretty simple. It's captured by this very simple schematic. So what I'm showing you here is this very simplified representation of the state of your brain. And you can think of these brain states as exhibiting these slowly changing fluctuations. And these fluctuations are affected by who you're seeing or what you're, where you're walking, for instance, or even what you're thinking about. They form both a spatial context and a temporal context, but they also form an internal context. Now, suppose that you and I engaged in a great conversation in a restaurant, and you want to remember this conversation. Well, if you're going to remember that conversation, you're actually going to embed the memory of that conversation within this context. Now, suppose at a later point in time, you're walking down the street and you happen to walk into the same restaurant. And when you walk into this restaurant, it triggers you to remember this conversation that we had. Well, there's substantial behavioral and psychological evidence that suggests that when this occurs, you actually mentally and internally jump back in time and you replay that conversation in your head, but you also replay the surrounding context. You might think about what you were wearing at the time, where you're going to next, and what you were thinking about. And so this idea of mentally going back and replaying an episode, it was coined as mental time travel by Tulving in the 1970s. And it presents us with an immediately testable hypothesis, which is that if there is a pattern of activity that is present in your brain when you first experience an episode, then when you go back in time and you retrieve that episode, you should actually replay a very similar pattern of activity. And so one of the first aims in our lab, or one of the first areas that we focus on in our lab, is actually trying to understand the mechanisms that underlie this process of replaying these memories. Now, what we also know is that this brain state has a significant impact on your ability to actually form and retrieve these memories. For instance, you might be more sleepy, you might be more awake, you might be paying attention, you might be paying less attention. And so another area of focus in our lab is actually understanding how these different brain states actually affect our ability to form and retrieve memories. And then finally, as I said, one of the main purposes of having a memory is to actually use these memories to guide our future actions. And so one of the other areas that we focus on in our lab is trying to understand how this actually occurs in the human brain. So today I'm going to largely focus on this first area, this idea of understanding these mechanisms that underlie human replay. And the experimental platform that we use to actually test these questions is actually taking advantage of the opportunities that we have through the clinical care that we provide in patients with drug-resistant epilepsy. So I'm not going to talk about epilepsy very much, but as some of you probably know, patients who have drug-resistant epilepsy continue to have seizures despite best medical therapy. And in those cases, we can actually offer them a surgical resection to actually remove that part of the brain that's causing seizures. But to do so, it critically relies upon our ability to actually localize exactly where those electrodes are in the brain. And if we can't do this non-invasively, we actually have to place these electrode contacts directly in and on the brain to be able to do that. So I'm going to show you a picture of a brain on the next slide, so just brace yourself. What I'm showing you here is a typical implant that we have. And so what we're looking at here is a temporal lobe in one of our patients. And you can see that we place these electrodes directly on the surface of the brain. There are also electrodes that lie underneath the brain. There are electrodes that go to the frontal lobes and so on and so forth. And we use a variety of different electrode combinations depending on the actual clinical focus that we're trying to localize. Well, it turns out that this gives us a very good opportunity from a research perspective, because while these patients are being monitored, they are awake and they're interactive. And so we can ask them to participate in different types of tasks or games, and we can record neural activity and ask how this activity correlates with the behaviors that we observe. And so from these electrodes, we can get a variety of different signals. So for example, from any one of these contacts, we can capture the net aggregate activity over a small patch of cortex. Just generally reflects the net dendritic inputs over 100 or hundreds of thousands of neurons. And what we get from this signal is something that we call an intracranial EEG signal. And what you'll notice about these intracranial EEG signals is that they are characterized by the presence of these fluctuations or these oscillations. These oscillations come in a variety of different frequencies. There's been a substantial literature that's linked different frequencies to different types of behaviors. And so this is one of the main signals that we observe. What we can also do is we can actually ask how signals might be correlated or coordinated across different brain regions. And now this begins to give us a sense of what's happening at a larger network scale, how information is moving across from one brain region to another. And then finally, with more recent technologies and more recent electrodes, we can now actually capture the activity of individual neurons. We can actually capture spiking activity and ask how individual neurons, how population spiking activity might be related to this process of forming and retrieving memories. So like I said, what I'm going to focus on today is what are these mechanisms that we've been able to uncover that mediate our ability to actually go back and replay these memories. And what I'm going to show you first is some previous work that we've shown where we've seen that there are patterns of activity in the brain, both at these larger spatial scales as well as these smaller spatial scales, that are reinstated when you retrieve these memories. And then we're going to ask how these patterns are actually reinstated, the mechanisms that underlie that. And I'm going to show you some data that tells us that there are actually sequences of spiking activity in individual neurons that are actually being replayed. And then finally, I'm going to make the argument that what this is telling us then is that these packets of information are really captured by these sequences of spiking activity. So the experimental paradigm that we use for these set of tasks is what's known as a paired associates verbal episodic memory task. So in this task, what we do is we present our subjects with these pairs of words. These are just regular nouns. We then ask them to make these novel associations between them. They then perform some simple math problems as a way of distraction. And then later on during testing, we cue them with one word, and their job is to remember what the associated word is. And we do this, we use this paradigm because it offers us two major advantages. So first of all, most of the electrodes that we place are actually in and around the temporal lobes. And these are regions of the brain that we know are heavily involved in semantic processing. In order for you to actually complete this task, it requires you to draw upon the meanings of these words to make these associations. And so what that suggests then is that the patterns of activity that we see in the neural data might be related to semantically processing the meanings of these words, and therefore might be relevant to the experimental questions that we're trying to ask. But the second and more important advantage is that this task gives us direct control over what word pairs we're trying to remember, or trying to encode its memory, and importantly, what word they're trying to remember and when they're trying to remember it. And so what that means is that we can look at what the activity looks like when people are forming these memories, and we can ask whether similar patterns of activity are actually being replayed when people retrieve these memories. So in earlier work, this is exactly what we did. So in one of our first studies, what we did is we looked at these patterns of oscillatory activity across these clinical surface electrodes. And so from each of these electrodes, we can extract out spectral power within different frequencies. We can aggregate all this together across all of our spatial locations, and we can generate a feature vector. And we can ask how similar these feature vectors are between when you encode a memory and when you retrieve it. And what we found across all our subjects is that we can create what we call these similarity maps. And so just to walk you through this, what we're looking at on the x-axis is every single time point during retrieval until the time that they vocalize their response. On the y-axis is every single time point during encoding, beginning with when the word pairs come on the screen. And the color of each pixel tells us the extent to which that feature vector is similar between that encoding time and that retrieval time. So if we see a big red blob, what that tells us then is that there is a pattern of activity that was present immediately before people vocalized their response that was quite similar to the patterns that were present when they were encoding these words into memory. Now, if we look at what happens during the incorrect trials, we find that this similarity largely disappears, and there's a significant difference between the two. Now, in subsequent work, we actually extended this down to the level of individual neurons. And so to do this, we actually use these special microelectrode arrays. These are colloquially known as Utah electrodes. They are arrays of approximately 96 electrodes, and they extend about 1 millimeter into the cortex. And what we do is we place these arrays directly into the anterior part of the temporal lobe. We place it in this region because this is largely an area that we anticipate we're going to resect. And each of these electrodes can capture the activity of individual neurons or action potentials. And now we can ask what this population of neurons is doing as people participate in this task. So just to give you an example, here is a population of neurons and their responses during the encoding period. You can see that when the word pairs come on the screen at time 0, there are neurons that show increases in their responses in red and other neurons that show decreases in their responses in blue. Now, if you look at what these same neurons are doing during retrieval, what you find is that they exhibit a very similar pattern of activity. That the neurons that were increasing their activity during encoding are also increasing their activity during retrieval. Those that were decreasing their activity during encoding are also decreasing their activity during retrieval. And so once again, we can construct these feature vectors. Now we can populate this feature vector with the population spiking activity. We could generate these similarity maps. And again, across all our participants, what we see is that there's significantly greater similarity during correct retrieval compared to incorrect retrieval. So together, what these data tell us then is that at both the local scale as well as at the neuronal scale, that when you retrieve these memories, you actually reinstate these patterns of activity that are particular to the individual item that you're trying to retrieve from memory. And so the question that's motivated us recently then is, how does this actually work? What is the mechanism that underlies this process? And so to do this, we actually drew a lot of inspiration and motivation from the literature on spatial navigation in rodents. Of course, everyone here is mostly familiar with this literature, but this is a quick summary of that work. And what I'm showing you here is a phenomenon that has been described in the literature for about 40 years. This is the presence of a specific type of cell in the medial temporal lobe known as a place cell. As was mentioned earlier in the earlier talks, these are cells that respond to specific locations, such that if a rat is moving along a track, what you'll see is a sequence of firing of these cells as the rat is moving along the track. Now, remarkably, when the rodent is asleep and perhaps consolidating their memory, or when they're awake and maybe thinking about different runs on the track, what we find is that there are the same sequence of spiking activities actually replay, suggesting that there's some element of memory that's actually involved in this process of replaying this sequence of spiking activity. Now, what's been less clear is what happens in other regions of the brain, like in the cortex, and certainly what we have not known is how such spiking replay might be relevant for awake human memory retrieval. Now, what we've also seen in these rodent studies is that when we see these replay events, they're often accompanied by another physiological signal known as a sharp wave ripple. So these are fast oscillations that are observed in the local field potential signal. They are called sharp waves because they oftentimes sit on the deflection of these sharp transients. In the filter trace, you can see that these are these fast oscillations. And these ripples have really come to represent or be seen as a biomarker for these replay events. Now, again, these ripples have largely been studied in the medial temporal lobe, oftentimes during sleep. Less frequently, we've seen and we've observed data showing how these ripples might be present in the cortex. And certainly, there's been less data examining how these ripples might be relevant for awake human memory retrieval. So we decided to tackle these two questions. So the first study is a study that's carried out by Alex Vaz, who's an ascending graduate student in our lab. And what Alex wanted to first ask is whether we could see such evidence for these ripples in our human recordings. So I'm showing you here on the left is a schematic from one of our implants. You can see that we have electrodes lying underneath the brain, right along the medial temporal lobe. And we have other electrodes sitting on the lateral temporal cortex. And if we look at one of these medial temporal lobe electrodes, what we can see in the raw trace above and in the filter trace below is the presence of these ripples. Now, if we look at the same time at one of these recordings from the lateral temporal cortex, in this case, the middle temporal gyrus, we can also see on occasion that we see these ripple oscillations. And at time, these ripples are actually happening at the same time between these two structures, so we call these coupled. And at times, they're happening in one region and not another, and so we call these uncoupled. And so we can see these ripples all the time. So now the question is whether these ripples are actually relevant for this process of memory retrieval. So what we're going to show you here is the data from one participant across all their trials. We're looking at what happens during retrieval. And what we're doing is we're placing a tick mark every time we see one of these ripples. And so what we can see in the medial temporal lobe is that during the correct trials, there's a clear increase in the number of ripples that we see immediately before people vocalize their response. This is much less during the incorrect trials. But importantly, what we also see is that there's a significantly greater number of coupled ripples during the correct trials compared to the incorrect trials. And of course, if we look at other parts of the brain, like the primary motor cortex or the primary sensory cortex, we don't see these changes. And we'd expect that since this task doesn't really involve those regions of the brain. So this is providing us some evidence then that these ripples, and particularly these coupled ripples, might be relevant for this process of memory retrieval. Now, what we also did is we wanted to tie these data to the data that we had seen earlier regarding reinstatement. And so what we did is we locked our retrieval data to every time we saw one of these coupled ripples. And what we find is that if we lock the data in this manner, we can see that these ripples, particularly these coupled ripples, actually precede this increase in reinstatement that we observe across the cortex. Whereas when we pick random times during the retrieval period, we see that that reinstatement and that locking largely disappears. So together, these data really show us then that these ripples and this interaction that we know exists between the medial temporal lobe and the lateral temporal cortex might be playing an important role in this process of retrieving memories and reinstating these patterns of activity. Now, what we want to do next then is actually ask what's happening during these ripples, and in particular, what's happening with the spiking activity underlying it. And again, we can do this because we have this ability to record from these populations of spiking activity through these microelectrode arrays. And so this is a second study that Alex carried it out. And what we wanted to know is what neurons are doing in the presence of these ripples. So what I'm showing you up top are these ripples that we've recorded on these electrodes in the middle temporal gyrus. So these are electrodes that are sitting on the cortex. And then we can also see that there is a ripple that happens slightly earlier in the medial temporal lobe. And if we look at what this population of neurons is doing during this ripple, what we find is that there is a clear burst of spiking activity. And in fact, if we look at the individual microelectrode channels, we can see that there are small little micro ripples on each of these LFP traces. And the extent to which we see these ripples on the local field potential signal correlates with how much spiking activity that we see. And we see this all the time. Every time we see these ripples, we see these bursts of spiking activity. And so we're in the process of characterizing this work. So this is work carried out now by another graduate student in the lab, Ai-Fong Tong. And what she's doing is she's simply asking, what does the relationship look like between these ripples that we observe and the underlying spiking activity? And you can see over this segment of time that whenever we see these ripples, both at the macro scale as well as at the micro scale and the local field potential signal, we see these bursts of spiking activity. As I mentioned, this correlation is quite strong. The extent to which we see ripple activity correlates quite strongly with how much spiking activity, both in the local field potential signal as well as in the intracranial EEG signal. Okay, so now what's happening during our paired associates task? So what I'm showing you here is an example from one individual trial. In this trial, we are presenting the word pair cake fox. You can see that there is a burst of spiking activity. And what we can do is we can actually ask which neurons fire earlier in that burst and which neurons fire later. And we're going to color those neurons yellow and blue respectively. Well, it turns out that when we present these word pairs, we actually see multiple bursts of spiking. And what's remarkable is that the temporal order in which these neurons fire is relatively preserved throughout each of these individual bursts. The yellow neurons always fire first, and the blue neurons tend to fire later. What's also remarkable is that that particular sequence of firing is specific to that individual trial. So what I'm showing you here is another trial. So in this trial, the subject was studying the word pair steam seal. We can see that the yellow neurons fire first and the blue neurons fire later. And now we're going to ask what these same neurons are doing when we look at a different trial. So this is a different trial. We've preserved the coloring. And what you can see is that now some of the yellow neurons are firing later and some of the blue neurons are firing earlier. The sequence has changed. And so we wanted to know whether we can actually sort of quantify this. And we can use any metric to do this, but the metric that we decided to use was a metric known as the matching index. So the matching index simply tells us how similar two sequences are to one another. And it's a bounded metric. So if the sequences are identical, the matching index takes on a value of 1. If the sequences are in the exact opposite order, it takes on a value of -1. And so what we're going to do is we're going to ask whether how similar sequences are within every trial compared to how sequences are when we compare across different trials. And so if we look at the trials that people eventually encoded correctly, what we find is that there is significantly greater similarity within the trial compared to across different trials. And we find that this relationship is actually largely absent in the trials that they got incorrect. And so what these data tell us then is that this process of successfully encoding these word pairs into memory involves exhibiting these precise temporal sequences of spiking activity that are preserved and consistent within each individual trial. Okay, so now the important question, of course, is what happens during memory retrieval. So what I'm showing you here is another example. Again, in this example, we are showing you the word pair Jeep Crow. We have this burst of activity in which the yellow neurons fire first and the blue neurons fire later. And now we're going to ask what these same neurons, preserving the same color scheme, are doing during retrieval. And so during retrieval, we present them with the word pair, with the word Jeep, and they say the word crow. And what we see is that we evoke a burst of activity that has a very similar temporal sequence. The yellow neurons are firing earlier and the blue neurons are firing later. And again, we can quantify the extent to which this happens using this metric, the matching index. And so what we find across all of our trials and across all of our participants is that as we get closer and closer to vocalization at time 0, we have these bursts of spiking activity that look very similar to the spiking activity and the sequence of spiking activity that were present during encoding. And this bar graph on the right here just sort of captures that difference during that last 500 milliseconds. Now, what we also find is that when this is happening, we also see overall decreases in spiking rates. And so what these data suggest then is that this process of encoding items into memory and retrieving items from memory involves sparse yet temporally precise sequences of spiking activity. So of course, we're quite excited about this work, and we're moving this work in multiple different directions. And so one of the areas that we're focusing on, of course, is how this might reflect representations of context, of time. And I'm not going to get a chance to talk about that today. But another thing that we're quite interested in is how this also might be relevant for how the brain represents individual items or content of memory. And if we can get a handle on how the brain represents individual items, then we can potentially track how these items are actually encoded into memory and how these items are potentially removed from memory. And the sequences that we see have motivated us to think about how the brain represents its information in a slightly different way. So I'm going to show you our two studies to get at that point of how the brain represents information using these bursts of spiking activity. And so in the first one, this is a study that was carried out by John Wittick, who's a staff scientist in our lab. And what we want to know simply is, how does the brain represent individual items? And so to do this, we looked both at intracranial EEG signals, as well as local field potential signals, as well as spiking activity, as individuals simply looked at pictures or read words that corresponded to different items. So they could be places, they could be people, they could be tools, they could be animals. And their job in this task was simply to tell us what it is, which category they're looking at, whether it's a person, place, animal, and so on and so forth. It's very similar to the localizer tasks that you see in fMRI studies. Now, the traditional way that you can do this, and the way that we have done this, is that you can look at a population of neurons and look at the population spiking activity. You can construct these feature vectors. And you can assign each of these feature vectors to each of these stimuli, and then you can build a classifier that can allow you to decode which feature vectors or what patterns are associated with which different categories. And we've done this, and this works quite well. But again, looking at this work earlier regarding our sequences, we've been motivated to look at this in a different way. And what we find is actually something that's actually quite interesting for us and potentially gives us some insight into how the brain represents information. So what I'm showing you here is a typical example. So if I show you, for example, a picture of Michael Jordan, what we find is after we present the picture, we evoke a burst of spiking activity that has a clear temporal sequence. The yellow neurons fire first and the blue neurons fire later. And again, when we show these images, we actually generally evoke multiple bursts of activity. And what we find is that the sequence of activity tends to be pretty consistent, that the yellow neurons tend to fire earlier and the blue neurons tend to fire later. And so once again, we can use this matching index, and we can ask how similar this sequence of activity is within trials compared to when we compare these bursts across different trials. And what we find in our preliminary data, and we've actually extended this now to 6 subjects, is that there is significantly greater similarity when we look within trials compared to when we look across trials. And this is true when people are paying attention, when they correctly categorize the item that they're looking at, and it's largely not true when they actually fail to do so. So this is actually quite exciting for us because it tells us potentially that this might be very important for how the brain might be representing information. From a computational perspective, this makes sense and aligns with some theoretical ideas regarding computational efficiency, because you have a much richer combinatorial palette if you can encode individual items using these sequences of activity. So finally, the final story I'm going to tell you then is related to the third sort of area of focus in our lab, which is how memories guide behavior. And we have a couple of projects where we are sort of working in this, that we are pursuing in this area. And so we have studies where we examine how prior knowledge and how prior memories might guide our ability to actually form memories, and might also guide our ability to search for memories. But the story I'm going to tell you about is how memories actually set our expectations. So imagine, for example, you're going back into this restaurant where we had this conversation, and you might notice that things have changed. You might notice, for example, in this case, that the restaurant is now black and white. And the reason that you've noticed this is that you had this previous expectation for what the restaurant should look like, and now what you're actually seeing is actually different than that expectation. And so the story that I'm going to tell you about actually builds on this emerging hypothesis in the computational literature that the brain is really optimized to maximize information. And the way it does this is it constantly generates an internal model for the way the world should look like. And then it compares how the world actually looks like to this internal model. And if there are any discrepancies, then those discrepancies actually carry information. We call those discrepancies prediction errors. And those prediction errors are quite informative for the brain. So just to give you a single example, imagine that you're walking along in this very nice field. You see a lot of grass, you see a lot of bushes, and you see a lot of trees. You don't really think much about the grass and the bushes and the trees other than they might be beautiful, but you don't really think much about them because these are the things that you'd expect to see in a field. Now, suppose out of nowhere up pops a television. Well, for you, this should be actually quite surprising and quite unexpected. And that unexpected event actually violates your expectations for this field. It is quite surprising. It is a prediction error, and it is therefore quite informative. This is, of course, the hypothesis of predictive coding, and this hypothesis, of course, has gained a lot of empiric support in the study of perception, in particular vision. So we know that you generate these internal models for how the visual world should look like, and you learn these internal models over a lifetime of observing and experiencing the natural statistics of the world. But what we're going to argue is that the brain is constantly making these internal models, and in fact uses memories to generate these internal models as well. And so the way that we tested this is in a study that we just completed. So this is carried out by Rafi Haq, who's another outstanding graduate student in the lab. And what Rafi did is presented our subjects with these images. These are images of these complex scenes. They have these associations between different items in the scene. And we present them these images once. And then later on during testing, we show them the same image and we ask them whether the image is identical to the image that they've previously seen. And so in many cases, the images are identical. But in some cases, we've actually manipulated the images. So in some cases, we've added items to the image. So in this case, for example, we've added this bird. In other cases, we've actually removed items from the image. In this case, we've removed this other bird. And the subject's job is to figure out whether the image that they're looking at is the same as their previous one that they've observed, or whether it's been manipulated. And if it's been manipulated, we then ask them to point to where that manipulation took place. And we do this also at the same time by tracking their eye movement, so we can see what exactly it is that they're looking at. And so actually to do this task, it requires you to actually draw upon your memory and to make a comparison between your memory of this scene, this internal model, and the scene that you're currently looking at. And so what we want to know is that if these two are in conflict, if there's a conflict between your internal model or your memory and your current visual experience, do we see evidence for a prediction error signal, and where does that actually occur? So let's look at what happens. So we focus largely on high frequency activity in these electrode contacts. And if we look at these different brain regions, what we see is an increase in high frequency activity that starts in the primary and secondary visual cortices, and then extends anteriorly to the visual association cortices, and then ultimately to the medial temporal lobe. And this is exactly what we'd expect. This is the ventral stream pathway of visual processing. You have this progression of activity from posterior to anterior parts of the brain. So this is what happens when we see an image that's identical to the image that we've seen before. So now what happens if we look at an image that's been manipulated? So if we look first in the primary areas of the visual cortex, we find that the patterns look quite similar. However, once we start looking in the association cortices, either in the temporal lobe or in the parietal lobe, what we see is that there's a clear difference in the activity. There is this elevated and prolonged activity in this high frequency band of activity. And then this activity, this change actually then emerges in the medial temporal lobe. And so for us, this actually looks like a signal that represents this difference, this prediction error. Now, if we look at the trials where people actually had an image that was manipulated, but they failed to identify that manipulation, we see the activity looks very similar to the activity that was present when they're looking at an exactly identical image. Now, let me also say that this is true whether we add an image to the item, whether we add an item to the image or whether we remove an item from the image. So it's not a property of the actual visual, it's not an artifact of the visual properties itself. So to us, this is actually quite remarkable. So you have seen an image in just one time. You have a single exposure, and this single exposure is sufficient for you to establish an internal model throughout your visual hierarchy about what this image should look like. And now you use that internal model to make a comparison to this new image that you're seeing. And what's also remarkable to us is that this change, this difference that we observe, arises first in these association cortices before it arises in the MTL. We have a lot of data in our manuscript here that sort of demonstrates these temporal differences. Now, what I'm also going to show you is that we can observe this difference when we look at this power, but we can also see this if we look at these discrete packets of information. So once again, we can filter our raw traces in this band, this ripple band that we observed earlier. We can identify these discrete time points where we see these bursts of activity, these packets of information. And so if we look at the activity of, for example, one of these electrodes during this task, what we find is that when we present these images that have been manipulated and the subject was able to correctly identify, we see a clear increase in the number of these packets of information, these little bursts of activity. And this is much greater than the number of packets that we see during the repeated trials or during the trials that were manipulated and people failed to identify correctly. And so putting all this together, what does this all mean? So what we think is going on is that when you first experience an episode or an event, as we said, you process information along this visual stream pathway. There are these interactions that we know exist between your visual association cortex and your medial temporal lobe, and it establishes a pattern of activity that represents that particular element or that item or that memory. Now, suppose at a later time you are presented with a similar but not identical image. You will first process this information again along the visual stream, and then through these interactions with the visual cortex and the MTL, you're actually going to pattern complete. You're going to reinstate those patterns of activity to retrieve that memory. But in these cases, what you'll find is that the patterns that you're reinstating are actually slightly different than the patterns that you're observing. And so this generates what we call a prediction error. And this prediction error first emerges in these association cortices and then is conveyed down to the medial temporal lobe, perhaps to update your memory for future experiences. Okay, so just to conclude, as I said, we use this general framework to study episodic memory. I showed you a number of different studies examining how we can use our approach to actually understand the mechanisms that underlie this ability to retrieve memories and to reinstate patterns of activity and to replay specific sequences of spiking activity in the human brain. I didn't talk very much about how brain states affect memory formation. I'm happy to do so if anybody has any questions that they're interested in exploring. But finally, I want to show you at the end how we can use this information to now understand how we use our memories to guide our future behaviors. And primarily, I showed you our work where we describe how we use our memories to generate an internal model for the world. And we use that model to make decisions about what we've seen and what we haven't seen previously. And so finally, I want to thank, of course, all of the members of my lab. They're all outstanding. These are the current members on the left and the former members in the middle, and all our very close collaborators on the epilepsy service and in the surgical neurology branch at NIH. And of course, we are always open to postdocs and we're always recruiting people. So if anyone's interested, please feel free to reach out. Thank you.
Okay, thank you so much, Kareem, for this fantastic talk. There are many questions in the question tab. It seems that maybe not very, it's a conflict of interest, but the upvoted one is mine. I'm just going to go ahead and read it quickly. So similarities in the activity patterns between encoding and retrieval are obviously visible with the spike rate activities that you've shown us, but also they're visible in the population level oscillatory activity, right, in the LFP signals. And so just curious whether what relationships or similarities you found between the spike rates. Are they more similar to gamma range population activity or more similar to theta or other frequencies in the different experiments you've seen? How do these two levels of observation relate to one another as it relates to these memory tasks?
Yeah, that's a very good question. So that's actually the work that we're trying to finish right now, where we are trying to characterize the relationship between the bursts of spiking activity and the overlying changes that we see in the EEG signal. I'm trying to bring up that slide right now so I can show you. But basically, the short story is that every time we see these bursts of spiking activity, they're accompanied by these ripples, these sort of fast oscillations in the 80 to 120 hertz band. And you can see an example of that right here. So you can see here on the bottom left, we have these clear bursts, these increases in spiking activity. Every time we see that, both in the local field potential channel, so on each one of these microelectrodes in purple, as well as the overlying intracranial EEG signal, these larger clinical contexts, we see these fast oscillations. So we think there's a clear relationship between this relatively fast frequency band in the EEG signal and these underlying bursts of spiking activity. And in fact, the contention that we are probably going to make is that these changes that we see in these high frequencies are actually punctate sort of discrete points of change. They are discrete time points that underlie or that reflect this underlying burst of spiking activity.
Okay, yeah, that I think that also agrees with some other data out published in literature showing these close correlations with the higher frequency rather than with the lower frequencies, right? So the broadband, but also, as you say here, the ripple activity more specifically. So that's interesting. We don't have much time. There's a panel discussion afterwards. I'm just going to take one last question. This is a question by Shahab Bakhtiari. And so his question is, how can one distinguish between episodic memory and short-term memory in the paired association task used in the study? The follow-up is it even necessary to dissociate different types of memories that might be involved in this task? So
yeah, so that's
short-term versus episodic.
Yeah, so those are two very good questions. So number one, to answer the second question, I would actually make that same argument, which is to say that while we are framing most of our work in the context of episodic memory and longer-term memory, it probably is relevant for shorter-term memory, and it's probably involving similar processes in that we will likely see replay of activity when you are retrieving a particular memory. The reason we think that this is not necessarily in the domain of working memory is for two reasons. Number one, when people perform this task, they have this encoding period where they study these pairs of words words, and then that's followed by a period of time where they actually solve these little math problems, and that lasts for about 30 seconds. And so no longer should these words be the focus of their attention. They should be focusing on the math problems. And so that serves as a sort of a form of distraction that then allows us to conclude that this is no longer in working memory. The other reason that we don't necessarily think that this is related to working memory is because there are limits to the capacity of items that one can retain in working memory. And in our task, we actually present these 6 word pairs, so that comprises 12 words, and so that should exceed the limits, or sort of what we believe are the limits of working memory capacity.
Okay, thank you so much for that answer, and obviously there'll be more time for discussion in the panel. So with this, I would like to thank you once again for this outstanding talk, and thank everybody else here. So what we're going to do now is we're going to have a shorter break than planned. It's going to be a 7-minute break, biological break, rest break, tea break, whatever you feel like, and we'll be back at 10:45. Hopefully that's okay with Dr. Nicole Russ. It's going to be a keynote lecture up. So please join us at 10:45. That's in about 7 minutes for the keynote lecture when Nicole Rust. And sorry? Oh yes, and just one quick mention, GatherTown space is open. So you can walk around with your avatar, go dance a little bit if you want to just have a small break on GatherTown before coming back. And there's a button at the bottom of the screen. If you see, there's a green button that says GatherTown. So feel free to go click on that. It will take you right to the space, and then you can go and explore. But don't get too excited. Don't enjoy it too much. We do want you to come back here at 10:45 for the keynote lecture. The space will stay open throughout the day, and we'll have more activities later. Okay, thanks once again. Thanks, Kareem, again for this outstanding talk, and we'll continue this in a few minutes.